Okay, so we've been talking the whole quarter, and I think if you go back and you look at all of the things that we've covered, as I did over the last couple of days, I think you're going to find that we've covered an amazing amount of information in this class, all the way from why clone genes to how genes are cloned to practical applications to what genes are, how they work, and all kinds of things. So it's been quite a remarkable experience. I'm going to give you some other insights into genetic engineering today and a week from today in things that you haven't really thought about. Some maybe a little bit, but I'm going to tell you about some very different things. And along the way, I'm going to intersperse it with some conceptual things that many of you have heard about vaguely, but I'm going to have to flesh it out a little bit more. And so this week and next week, just to give you a heads up, this week and next week will be the last science lectures that you will have in this class. And then for the remaining 4 weeks of the class, it's going to be all law, politics, political science, and things that relate to the impact of science on our society and all kinds of other things. It is working. My sound was off. There we go. You remember that, first day of class. Genetic engineering for insect resistance. So remember, this was taking a bacterial toxin gene and putting it in a plant and making the plant insect resistant. And we've talked all about almost everything that deals with that genetic engineering application. So there's the non-genetically engineered plants and there's the genetically engineered plants. And we discussed, and you had in discussion, how important that is in terms of being able to drive agriculture. So Albert, I'm going to come back to you. Tell me exactly how that worked.
How the genetic engineering...
Yeah, how did you see this genetically engineered plant, this beautiful time-lapse photography where we put a gene from a bacteria inside a plant and got it to work, and we stuck these worms on this plant and they just die because the plant makes the bacterial protein. How does that work? What did you have to do in order to do that? Just conceptually. Just bring it all together for us.
When we...
Where did the gene come from? How did you get the gene? How did you put it in the plant? How did you engineer it? Just give me some bullet points here.
All right, so we got the gene from the bacteria.
Okay. How did you make the library?
By taking it out.
What's it?
The gene. Or the library of the entire sequence of DNA.
So how did you make that library conceptually? How did you do that?
We just cultured the bacteria.
Yeah, how'd you make the library, though? How did you make a library of the genome, Albert? This is old hat. This goes all the way back to the first week of class. How did we do that? You with me, Dora?
Yes.
You remember how to make a library of the bacterial genome? How do you do that conceptually?
You take the, you have to do the transcriptase with the mRNA.
The what?
The mRNA.
Making a library of the bacterial genome? Whew. Man, when's this exam? On Thursday? What do you think, Lauren? You want to help them all out? I thought this was a giveaway today.
Could you extract the chromosomal DNA and then use restriction
Well, you
enzymes?
tell me. I'm not, you're asking a question, and I'm asking you the
Okay,
question.
you could extract the chromosomal
You
DNA.
could?
or
That's-
You extract the chromosomal DNA out of the bacteria and then use restriction enzymes to cut it into the pieces, and then you could put pieces of the DNA into plasmids and
Okay,
and let them multiply.
so you made a library now. Okay? How'd you find this toxin gene in that library? That's a hard question. Well, how do you think you found that gene?
probes.
How would you get the probe? This is all on your take-home test. Where would you get the probe from?
You would have to have a sequence
A sequence of what?
of the gene,
Well, the point is, if you're trying to find the gene, you don't have the sequence of the gene.
right?
So you have to have a probe of some kind. So let's say you find that gene. Okay, now let's go back to a quiz that all of you signed your name off on. And how do you engineer it so that you can engineer it into the plant? Marvin, what do you think? How do you get that thing to work in the plant? What do you need?
You need restriction enzymes to cleave the,
What do you need to get that bacterial gene active in the plant?
a particle gun to shoot the
Active in the plant. That's fine. You can get it in the plant with the particle gun, yeah, but what do you have to do to get it to be active in the plant?
You have to incorporate the DNA into the plant DNA, the plant chromosome.
But what do you have to do, Faria, to get the gene active in the plant? Active.
Turn it on using the gene promoters or the promoters?
Absolutely. From where? The bacteria or from the plant? Marvin, did you forget about that?
I remembered as soon as you
Yeah.
re-asked the question.
Where would you get those promoters from, Faria? Would they come from the bacteria or would they come from the plant?
Bacteria?
You mean you're going to use a bacteria promoter to express the bacterial gene in the plant?
No, in the plant.
Where are you going to get those promoters from? From the plant. What would you have to do to the plant genome then to get them?
Sequence them?
Well, you could, but the sequence isn't going to give you the promoter. What's the only way you can grab it and have it in your hand for you? What's the only way that you can have something to be able to use it?
You need to have it?
Yeah, what's the title of this course?
Genetic engineering?
So how are you going to get this promoter?
Through genetic engineering?
Well, how are you going to find it? What is the feature of the genes that allows you to find that promoter? We covered this in a whole lecture in week 2.
Restriction enzymes?
Well, how do you know where the promoter is? What's the feature of it?
It has like a certain sequence.
Yeah, so is it at the 5' or the 3' end of the gene?
5'?
Yeah, so you think you can find it by its sequence, and then how are you going to get it away from the rest of the gene?
By adding it to enzyme restriction enzymes?
Well, how are you going to know where the restriction enzyme sites are? Once you've cloned the gene, how are you going to know? I think that's on your take-home test too, isn't it? It's a little restriction map on there. Have you got to that point in time?
Not yet.
Oh boy, you're going to be up for the next 3 days.
Yeah, I think so.
I think so. You know, it's almost impossible to do this exam. If it's not done by now, it ain't ever going to get done for you. What do you think about that? Huh? Anyways, you got to use restriction enzymes. The point here is, I put this up here because I thought by now all of you would have been able to synthesize a lot of these different things and bring it all together, because that little film clip exemplifies all the different things that we've discussed in discussion and in lecture and on your quizzes and on your take-home tests, because it really means going from the gene, understanding what the gene is in bacteria, how you isolate the bacterial gene, how you find it, how you then use that bacterial gene and engineer it with parts of the plant gene, because we talked about the modular organization of plant genes and bacterial genes and splicing things together. I went into that in long length. And then you take that promoter, and if you remember in discussion, I think it was either week 3 or week 4, you had a quiz on taking the switches from the plant and fusing it to the bacterial, toxin gene essentially, and putting it into the plant by using genetic engineering, which you had a whole discussion on, and you're correct, Marvin, in terms of the particle guns. So the idea is to be able to, what Dr. Harada and I want you to be able to do is to synthesize all of these things and to try and bring them together. And hopefully on Thursday you'll be able to do that. In any case, we've talked about a remarkable amount of things relating to genes and how we use these things. And remember, I showed this on the first day of class, so I'm going backwards, that the reason why you clone genes and the remarkable revolution of genetic engineering was that you can isolate genes and, Faria, get them in your hand. That's why you used recombinant DNA, because as all of you know, you can't isolate a gene apart from another gene without using recombinant DNA. Of course, now you know you can use PCR, but you have to have sequenced and cloned the gene before, in fact, you can use PCR. But the point here is then you can use it for a variety of different applications, some of which we have discussed in class and in discussion, and many we have not. And as I've said, and we're going to come up to this over the last course of this course, genetic engineering has affected you, your lives, my life, society in many, many different ways, and has allowed for many new observations and discoveries and applications, some of which are written up here, the biotechnology industry, understanding your own genome, understanding where we've came from, all the exciting and fun things that we learned, and many, many, many more. So all of you know these kinds of things, but hopefully I'm going to get this thing to work. So that was the beginning. And we talked about that in discussion. There's a lot of myths in this little clip, particularly the frog gene was not really a gene that encoded proteins, but I think you get the point. The point is going backwards for the invention of this technology, going forward to how it's now been implemented and how exciting it is. And in terms of the last part of this course, we'll be talking about the kinds of laws and legal aspects which allowed the guidelines to be established. So the point here is that what genetic engineering has done more than anything, and we've discussed it in this class, is to put forward basic knowledge. That's the most important thing being able to clone, manipulate, and study genes has done, and we've talked about that. Being able to look at genes, being able to understand the structure of genes, such as the fact that they have introns, being able to understand what the proteins are that are encoded by these genes, being able to study the genome, the study of genomics. All of these different things led to an enormous amount of basic knowledge. And so I would say here, as you can see with all of these books, as I said many weeks ago, probably 80 to 90% of all the information in these books has been discovered because of the ability to clone and study genes, whether it's developmental biology, human genetics, genomes, which is a whole new field allowed by genetic engineering, biology, molecular biology, cell biology. All of these things in these books have been allowed because we can isolate genes, understand what they do, understand what the processes are in the cell, pick them apart by a variety of different procedures. And clearly then, as you can see here, you can find out all kinds of remarkable things. And in fact, that's what I illustrated with this jellyfish experiment, the glowfish, because at some point someone, in fact, a guy by the name of Martin Chalsky, who won a Nobel Prize last year for being able to use this green fluorescent protein in biology, but clearly one had to clone the green fluorescent protein, the protein that makes the jellyfish Green, someone had to determine the structure of the protein. There it is. That was done after it was known what this gene and what this protein was. And you can see that right here is a little chromophore, which is the structure of this little molecule here. And that's exactly what makes this little protein fluoresce in the jellyfish in the ocean. But now you know you can take this out by using this genetic engineering. We've discussed this ad nauseum. Add switches, add different kinds of things, put it in mice, put it in fish, put it in plants using this experiment. And there's the experiment that Cohn and Boyer carried out that you read about the first week of class, and you just saw a film clip. So clearly, the major application of genetic engineering is just simply, and it's not simply, basic knowledge. Anything you learn in biology today certainly came from genetic engineering. And then we discussed many of the applications that could be used because you have a specific gene or you have a specific genome that you can study. And we talked about fingerprinting, which we've done in the class, talked about following disease genes, making super mice, human genetic variation, which I talked about last week, putting disease-resistant genes into plants, which I just showed you the clip, engineering humans, which I'm going to talk a lot about next week, and of course what you're going to talk about tomorrow, and I'm going to talk a little bit about next week, engineering animals. And so there's been some applications that we've talked about in limited ways in using this technology to do some remarkable stuff. We've talked a little bit about making proteins in bacterial cells. I'm going to talk more about that today. We've talked about using genes to diagnose and screen for human diseases. We talked about forensic applications. We talked about genetically engineered plants, and so on and so forth. But there's many, 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 many more applications of this technology. There are some of them right up there, of which the list is almost too long to be able to discuss in depth all of them in this class. But the ones in black will be ones that I'm going to be discussing over the next couple of weeks, this lecture and next. The ones in blue we've discussed already, and there's no reason to go into them again, but it's an amazing collection of applications. So I want to start with genetic engineering applications using bacteria. I'm going to talk about drugs. I'm going to talk about vaccines. I'm going to talk about antibiotics. I'm going to talk about cancer. I'm going to talk about bioremediation. I'm going to talk about all kinds of ways that bacteria can be used and genetic engineering for some remarkable things. And then I'm going to switch to using molds in order to do that. And then I'll probably quit today because it's going to probably take me that long. And then I'm going to pick up on animals and plants next week. So let me remind you about bacteria. You know that they're these simple organisms. They're actually not very simple, but we've talked about them many, 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 many times. But they have a lot of diverse effects on humans, on plants, on the environment, on a variety of different things. These are the ones that are used for recombinant DNA, E. coli, but in some respects, as I'll show you, E. coli can be a very, very serious pathogen. You can see bacteria that are involved in the nitrogen cycle, cycling nitrogen. You can see that bacteria used for antibiotics. There's the bacteria that grows in the hot springs. From that bacteria, the DNA polymerase, which is used for PCR, was isolated because it's stable at 85 degrees. There are some bacteria that are involved in human pathogenesis, a variety of different things. Many, many bacteria cause a variety of human diseases, but they affect all kinds of other things on the totality of the face of this earth. But from the point of view of genetic engineering, we're really focusing in on E. coli. And as you know from week 2 in discussion and from your take-home test and a variety of different things, one has to be able to clone a gene that may have some importance, let's say, in terms of making a drug in humans, and I'm going to review insulin in a second. And clearly you have to have a vector, and in this case you have to have what we call an expression vector. I touched upon this when I discussed factor VIII, and the expression vector means that it has the switch, which allows the gene to be active inside a bacteria. You can then place some kind of foreign gene after that switch. In this case, it's a cDNA or a synthetic coding sequence, as you will see, because you don't use the genes because they have introns. You can then put that in the bacteria. You can then scale that up enormously in fermenters, as I'll show you, and you can make some foreign protein, let's say insulin, TPA. You can make growth hormone, a variety of different things to use them for, let's say, medicinal purposes. Now, what are the reasons for using E. coli as a vehicle, a factory to make drugs or to make proteins, not simply drugs, but in this case, making drugs? Because we know how it works. It's the workhorse of genetic engineering. We can do all kinds of things with it. You can make an unlimited amount of protein. You can engineer it in any way. You have these plasmids that you can get in and out. So you can do all these things because there's been so many years of genetic engineering and bacterial genetics and all kinds of things that have been done with it. But it has a tremendous disadvantage. You can't make all drugs in E. coli because remember I said sometimes some human proteins have to be modified after they're made, like factor VIII, in order to get it to work properly, and E. coli doesn't do that. And from that point of view, you use either a mammalian cell, you engineer a mammalian cell, or yeast, as you will see. But clearly, many drugs, as you can see, are made in E. coli, some in yeast or mammalian cells, and those in yeast and mammalian cells, again, are used because they can modify the proteins like human cells can modify the proteins in order to get them to work. Tiger, do you understand that? You sure? Samantha, you understand that?
I have a question.
Go ahead.
Okay. How would you necessarily modify the proteins?
I'm not modifying it. The cell is modifying it.
So not all proteins made in E. coli will actually work in humans?
That's correct. We've discussed that in the context of factor VIII. You remember that lecture?
Yes. How would you modify them after?
I'm not modifying them. The cell is modifying them.
The cell modifies?
So the U is not the correct noun.
How would the cell modify?
It adds, remember I said if you go back and review your factor VIII lecture, we discussed the fact that factor VIII has little sugar molecules put on it, and those sugar molecules are important for its function, and that if you don't have those sugar molecules on the protein doesn't work, and E. coli can't put those on because it just doesn't do it. But a mammalian cell, which if you go back and review that lecture, which was the last part of that lecture, if you engineer mammalian cells to make factor VIII, it adds those sugars because that's what it does normally like human cells. Okay? So again, though, bacteria make a lot of drugs, and there's a lot of reasons for it. And one of the ones, which is the classic that you had in discussion, but it was the first drug made by genetic engineering, and it was the one in which the biotechnology industry was established, was insulin. And as you know, and I'm only going to go over this briefly, insulin is required to regulate your sugar metabolism in your blood. And in fact, in the pancreas, you know that there's a couple of cell types, these beta cells and these alpha cells in the islets of Langerhans, and it's in the beta cells that the insulin gene is activated. And you know that the reason why the insulin gene is only activated in the beta cells is because, Katie, the insulin gene has the switch for activation in the beta cells. In other words, those activators on the upstream region of the insulin gene will only be active in these cells because there's proteins, and we've discussed this in the context of the I protein and others, there's proteins that interact with the activators in the upstream region of the insulin gene to activate it only in that cell. And furthermore, when insulin is made, it's made as one protein. And this is a little connector that connects this chain and that chain together. In other words, there's one insulin gene that specifies one insulin messenger RNA that specifies one insulin protein. Here is the amino acid sequence of that protein, which was the first protein that was ever sequenced by a guy by the name of Sanger, who discovered the method for protein sequencing and won a Nobel Prize, and also discovered the method for DNA sequencing and won a Nobel Prize. And so he's quite a remarkable guy, this Fred Sanger. But this was the first protein that was ever sequenced because it has a very, very, very small number of amino acids. When you isolate it, it looks like it has two different chains, an alpha chain and a beta chain that are linked together by these bonds here. But in reality, it's one protein, and after translation, Samantha, that protein is processed after translation, and this little red thing is digested away. Okay? And so it looks like it has two chains in the cell, but it's made as one chain, and then it's processed. That processing occurs inside the beta cell of the pancreas. And then, of course, this little hormone is secreted from the pancreas into the bloodstream, and if you eat a candy bar or if you drink a Dr. Pepper, what happens is your sugar level goes up. If the sugar level goes up, a signal goes to secrete the insulin. The insulin goes into the bloodstream, and there it is, and it binds to what's called the insulin receptor. It binds to the insulin receptor in the liver. And when it binds to the insulin receptor in the liver, a signal is sent. There's a little receptor, and we're going to come back to this later on in this lecture in a different context, but there it is on the membrane of the liver cell. Here, in fact, is the receptor. A little signal is sent, and notice that signal is sent to this little response protein right here. And again, this is like a cascade. Remember I talked about the cascade of clotting inside the blood? It's a slightly different kind of cascade, but clearly the insulin binds to the outside of the receptor, sends a signal. That signal goes to the inside. There's a little protein. A phosphorus is added. And when that phosphorus is added, that protein then interacts with an enzyme called glycogen synthase, modifies it, and says you're supposed to be active now. It was previously inactive. In the absence of insulin, this enzyme was inactive. It becomes active, and then it takes glucose from that Dr. Pepper or from your candy bar, and it binds them together into glycogen, big long carbohydrate polymer of sugars, and stores them in the appropriate place. And then, of course, when you start starving, the reverse occurs. Insulin levels go down. When insulin levels go down, another hormone that's made in the alpha cells called glucagon goes up. When glucagon goes up, the reverse occurs, and glucagon essentially is involved in activating a different enzyme, and that enzyme acts on glycogen and releases the sugars from glycogen, so you can use those sugars into the cycle that makes energy involving the mitochondria. And so insulin is a remarkable hormone that evokes a signal pathway. It's called signal pathway, which it binds to this receptor in a site where it's not made, in the liver. That's not a site because the insulin is made in the pancreas, and then that signaling activates this enzyme. That enzyme is inactive in the absence of insulin, and that enzyme takes these sugars and binds them together, makes them into big polymers, glycogen, and stores them. And if you have a defect in either the binding of the insulin or a defect in the production of insulin, mainly you can't produce it in the pancreas, you have diabetes, and it's a pretty serious disease and nothing to take for granted. And that's essentially how that works, and here's the scheme over here. So clearly one can clone the insulin message from the pancreas, as you know from discussion. And by cloning the insulin message, you can then get it to be expressed in bacterial cells, and you can make insulin. But that's not how it was done. And that's not how it is done, as you may remember from your discussion. When insulin was first made into a drug using recombinant DNA, insulin was synthesized using the genetic code and using the amino acid sequence of the alpha chain and of the beta chain. By knowing the amino acid sequence, Max, you can get the sequence of the cDNA, right? Just look at the genetic code and just get the sequence. Of course, there's this thing called redundancy, degeneracy in the genetic code, but it can be done. And so the way in which insulin is made and has been made for 30 years was to simply synthesize organically in a lab. We're not talking about using biology. We're not talking about enzymes. Marvin, we're not talking about PCR. We're talking about taking each nucleotide and having those nucleotides being bound together, phosphodiester bonds, one by one, in the precise order, 5' to 3', and the A chain and the B chain, alpha and beta chains, were made separately. They were synthesized, cloned into separate plasmids, put into separate bacteria, A chain, B chain made separately, purified in remarkable ways, which I'll talk about in a second, and then combined together after they were made into recombinant insulin. So the point here is that we have synthetic A chain, synthetic B chain. Those are made in bacteria. The A chains and the B chains are then put together after they are made and purified from bacteria. And there's elaborate fermentation plants in which these bacteria are grown. This is scaling up the process in these fermenters, growing just liters and liters and liters and liters and liters of bacteria and getting them to synthesize the proper proteins that they're supposed to synthesize, and then purifying these proteins using elaborate purification methods so that they can be very pure, meaning on this gel electrophoresis of proteins, not nucleic acids, you see a very pure protein and nothing else. And then, of course, you can finally make this humulin, which is the recombinant form of insulin. And then, obviously, in order to be able to get this to be done and used for human consumption, you have to have approval by the FDA. You have to go through an elaborate process. We'll talk in a couple of weeks of what the legislation is that allows that to be done, but once it's approved, you can use it as a human drug. And so you've had all of that, but it's an amazing advance. But clearly, many, many, many drugs are made in that way in a huge process, and they've done a tremendous amount for humanity in terms of being able to treat disease. Okay, now, what you don't know is that the same kind of procedure is used in a variety of ways using recombinant DNA and genetic engineering to make vaccines. Why to make vaccines? Because, you know, you'd like a diphtheria vaccine, you'd like a vaccine that can combat all of these different pathogens that you can see up here. All of you take typhus, diphtheria, variety of different vaccines, hepatitis A, hepatitis B, hepatitis C, smallpox, which, how many of you have been vaccinated against smallpox? Anyone in this room been vaccinated against smallpox? Amelia, you have. I'd bet my life on it. I have. Anybody else? It's pretty amazing, since this is one of the biggest scourges that has ever been on the face of this earth. It's remarkable that you don't get vaccinated anymore against smallpox. Why? Because it's been eradicated from the face of this earth. It's the only disease that's been eradicated from the face of this earth, completely. But we used to have smallpox vaccinations almost immediately when you were born. But the point here is here's a whole series of bacterial diseases. Here's a whole series of viral diseases, smallpox included. But look at them. Ebola, a variety of different things. Measles, you've been vaccinated against measles. Polio. How many of you have been vaccinated against polio? Oh, I'll bet you most of you have. I'd be betting on it. Max? Amelia has, I have. Polio. Anna, tell me about polio. Did you ever hear of something called polio?
What do you want to know?
Did you ever hear about polio? Do you know what it is?
I've heard about it.
Tell me what you've heard.
I'm pretty sure we get vaccinated, right?
Tell me about polio. Know anything about it? Nancy, do you know anything about polio?
I think that it keeps you from breathing, and in the old days, you had to use an iron
The
lung.
old days.
Yeah, the old days.
Hear
you had
that,
to
John?
use
The
an
old days.
iron lung to breathe.
Keeps you from breathing, huh?
Yeah,
Is that all it does?
Cage can't go up and down.
Why?
Too much fluid buildup in the
Too
lung?
much fluid? Sure you, okay.
Paralyzes.
Yeah,
It paralyzes
you're
you, yeah.
paralyzed from polio. I think it's really good that most of you don't know about polio. I think it's fabulous that most of you don't know about polio. I can't tell you how many of my elementary school friends that are now my age can't walk because of polio. Because when I was a kid, we weren't allowed to go outside in the springtime because our parents were scared to death that we would get polio, because there was no vaccination to polio. And it was really a very serious disease, because if you got it, and if you got it seriously, like Nancy said, you got paralyzed so badly that you had to survive on an iron lung. And in fact, tragically, the last person ever to be on an iron lung died last year, who had polio and lived in an iron lung her entire life. A devastating disease, again, that was made, like, again, in the class here, you don't know what it is, something that you don't think about because of these things called smallpox. So smallpox vaccines, polio vaccines. I know all of you have been vaccinated against chickenpox. I know you've been vaccinated against measles. Many of you may take a flu vaccine. But the point here is that there are certain ways in which one can then combat these diseases. In any case, let's go back a few hundred years. More people died in the Revolutionary War to smallpox than died because of the battle. And in fact, George Washington had all of his men inoculated by taking a little bit of fluid from victims of smallpox and inoculated people in his army to be able to essentially vaccinate them, that is, elicit an antibody response to the polio antigen in order to be able to protect his army. And if you notice on this slide, in the 20th century, they estimate quite a few hundreds of millions of people have died from polio. And in fact, more recently, smallpox rather, has been wiped off the face of this earth. Now, all of you know the story of Edward Jenner, and I should say, going back, that smallpox was really the first, smallpox virus was the first biological agent used in warfare. People knew about it. People knew that it was infected. They used to take infected blankets and throw it over forts and throw it over, encampments and try and infect their enemies in order to win the battle, so to speak. About 20 years after George Washington inoculated his troops with smallpox, Edward Jenner discovered this thing called cowpox, which was a related virus in which he thought he could, was this milkmaid story that all of you know, that milkmaids were around cows that had cowpox, and they never, ever, ever, ever got smallpox, even during the most remarkable pandemics. And so what he thought was, but people had known this for a while. It wasn't that Jenner invented inoculation or vaccination, but it was that he used a cowpox virus. And the reason why it's good to use a cowpox virus is it is what we would call attenuated, meaning it doesn't cause a full-blown smallpox infection, because it's not really the smallpox virus. And so he inoculated people with this smallpox, this cowpox virus, and he found by doing experiments on real people, which we would never do today, that it protected them against smallpox. And he found that out by inoculating people with the cowpox virus and then re-inoculating them with smallpox virus from the lesions that were produced by smallpox. And he found that these individuals were protected from that particular smallpox virus. And so we have these things called vaccines. And vaccines, the word comes from vaca, or cow, because of cowpox, and that's where vaccines come from. Now, how do vaccines work? They work by eliciting your immune system to produce antibodies that previously existed. And I'm going to talk about why those antibodies previously existed. In other words, you are born, Kathy, with almost every antibody that you could ever imagine that could fight off almost any infection you could ever get, even before you even see those infections. Isn't that amazing? And if that wasn't the case, we'd all be dead. And I'm going to show you why that happens in a second. But the point here is that when you vaccinate someone, you vaccinate them with this antigen, which is a foreign protein, and it causes a process inside your body that stimulates the production of blood cells, so to speak, that have preformed antibodies on them, that people have been engineered to overcome that. We're going to talk about that next week. That is genetic engineering of humans to be able to overcome this genetic disease which destroys the immune system. These are the so-called bubble boys, and then to be able to overcome that by using gene therapy or genetic engineering. So here's how it works simply. Let's say you get a wound. A little stick goes in your skin, carries around bacteria. Those bacteria then are recognized, and a signal, again signaling, goes off. In your blood cells you have these cells right here, which are called phagocytes. This is only one aspect of immunity. Those phagocytes have antibodies on the outside of their surface. Those antibodies are specific for that particular antigen, which might be a bacteria. It crawls to the site of the wound. That's why it gets inflamed and gets red. It's because these phagocytes with these antibodies are crawling to the site of the wound. They essentially engulf these antigens and get destroyed in a particular way. And furthermore, you can see here's the bacteria, here's a phagocyte right here, here's a blow-up of that process. The cell has the antibodies exposed on the outside. Those antibodies recognize this bacteria. They basically grab it, engulf it, and destroy it, and clear it of the system, and then you're protected from the infection of that particular bacteria. The way it works in a nutshell, and remember we're on the march towards making recombinant vaccines, the way it works in a nutshell is you have these cells, and these cells develop when your blood system, your vascular system, and your lymph system gets developed. And these cells, which are called B cells, it's not important, each one of them has a different antibody, meaning each of these B cells makes a different antibody, and the potential number of antibodies that you can make is in the number of billions, billions and billions. And again, you're born with the potential, Derek, to recognize any foreign antigen that can ever be presented through your entire life simply because of a mechanism that I'm going to show you. And if this system is not developed in its proper way, it leads to death, such as SCIDS, the bubble boy, or in fact AIDS, that has the capacity not to destroy these B cells, but another cell, which I'm not going to talk about, which are called T cells, which are also involved in this kind of immune response. B cells make these antibodies and secrete them into your vascular system or your bloodstream. Is that clear, Maura? So you have antibodies because they're produced by these B cells. And each B cell produced is a different kind of antibody. The T cells that I'm not going to talk about are also in your bloodstream, but the T cells don't secrete antibodies. They're involved in engulfing things that infect them and other cells. It's a slightly different kind of immune response, and I'm not going to get into it, but the principle is the same. Here is an antibody. The antibody binds the antigen to a very specific location. These are made by these highly specialized cells. They're pumping out tons of immunoglobulin messenger RNA. If you wanted to clone an immunoglobulin messenger RNA, you'd isolate these B cells. You can see all these membranes here. These membranes are just making tons and tons of a very specific antibody. Brian, one B cell, one antibody. Every B cell makes a very different kind of antibody, and inside your blood system, you have the potential for making billions of different antibodies. Mike. Brian, oh, Stephen. Go ahead.
Antibodies are proteins.
Antibodies are proteins.
Which are coded
There it
for
is
by,
right here.
Which are coded for by genes.
Genes.
But if there's only a degree of tens of thousands of genes in the human
We're going to talk
body-
about that in 1 second, Stephen. That's a great question. That's exactly the question I sort of raised a few weeks ago. So how can you make billions of antibodies, billions of different antibody proteins, if you only have 25,000 genes? I'm going to answer that question in a second. Here's the antibody protein. It consists of a small chain called the light chain and a large chain called the heavy chain. Okay? The light chain is encoded by a light chain gene. The heavy chain is encoded by a heavy chain gene. Notice that there's 2 parts to each of those chains, a C and a V, a C and a V. The C is an amino acid sequence that's identical for all antibodies. The V is an amino acid sequence that's specific for each antibody. And so notice that this antigen, this specific antigen, binds to the V region of the antibody. It has to bind to the V region of the antibody because that's what makes the antibody protein unique. B cell. B cell makes specific antibody. The B cell has a light gene and a heavy gene. The light gene makes the light protein, and the heavy gene makes the heavy protein. The protein has a constant region and a variable region. The constant region is the same amino acid sequence no matter what antibody it is. The variable region is different for each specific antibody. Here's a specific antibody. It binds a specific antigen. It could be a bacterial antigen, a viral antigen, could be any kind of antigen, but it's very specific. Here are the B cells. These B cells are made during the development of you, from the time of conception to you become a baby in the mother's womb. These B cells are made when your blood system is made, and during the production of your vascular system, these B cells all get to be able to make different antibodies, even though they haven't seen any foreign antigens. And how does that happen? Because you have to have an antibody to everything that you could ever see during your whole lifetime, even though you haven't seen it when you're just a baby or being developed inside the mother's womb.